So my name is Serge Przedborski, and before I start my lecture, I would like, of course, to express my gratitude to the organizing committee for the kind invitations and for giving me the opportunity to talk to you today about some of the work we're doing in the lab to try to connect alpha-synuclein to the mitochondrial associated membrane protein as perhaps a new pathogenic mechanism of Parkinson's disease. As far as my disclosure are concerned, they're listed here, but there is nothing relevant to my presentation today. And these slides, it's really simply to remind us that basically the mitochondrial link to Parkinson's disease represent likely the most resilient pathogenic hypothesis of PD that came over the years. I'm sure that you know that in different flavors, ranging from deficit in bioenergetics to more recently with the discovery of Parkin and PINK1, the idea that perhaps autophagy of mitochondria or mitophagy was defective in the disease. And today what I would like to introduce is the possibility that perhaps there is a new flavor to the mitochondrial link to PD, and that is the associations of synuclein with the mitochondrial associated membrane. What is the mitochondrial associated membranes, or MAM in short? It's shown here on this electronic microscopy, where you have here a nice mitochondrion. Here you have the ER, and here with the red arrows, you see this nice ER membrane that is opposed to the other membrane of mitochondria, and that is the MAM. So if you look at the drawings instead, which probably make it a little bit easier to understand, here you have the ER, here you have the mitochondria and the other membrane, and you can see that part of the ER really opposed nicely along the other membrane of mitochondria. But you can also appreciate that this opposing part of the membrane is different from the regular ER. This specialized area is the MAM, and as you can see, it's enriched in a variety of proteins, including some tethering elements like mitofusin 2. And these structural and functional elements, MAM, is very important for a variety of function and collaboration with mitochondria that involve, for example, the biosynthesis of phospholipid, as well as cholesterol, as well as contributions to the calcium signaling homeostasis. And we'll come back on that later. Now that we know what MAM is, let me tell you how and why we decide to study synuclein in the context of MAM. And to do that, I have to go back to these slides that represent the salient point of a study that we did in 2002 with Bill Dower when he was still at Columbia University as a junior faculty. And basically what we discovered here and reported is when you delete synuclein in the background, in the genetic background of a mouse, you render the animal resistant to MPTP toxicity. And for years, people in my lab have tried to understand what could be the molecular underpinning of this resistance of MPTP in absence of alpha-synuclein. And that was basically really arduous work, and we never could understand why those animals were resistant to MPTP. Until more recently, where a series of elegant studies suggested that alpha-synuclein was accumulating either at or in the mitochondria. At the first glance, that appeared to me and probably many people that was in keeping with what we knew with alpha-synuclein, and that is that alpha-synuclein like lipid bilayer membrane and RAF-like domain, which are regions enriched in cholesterol and phospholipids. You have that in the synaptic vesicle, we knew that, but it happened also at the level of the MAM and the mitochondria. And so it came as a logical possibility for the intracellular localization of alpha-synuclein. However, at the same time, I was puzzled by these observations, because if you look at synuclein through a different algorithm that aim at identifying in given proteins a targeting sequence, which are those sequences in the protein combinations that allow a protein to be targeted to the mitochondria. We failed to identify any such targets. Therefore, what we did here is to try to clarify a little bit, basically more for us than anything else, whether synuclein can be imported in the mitochondria as a typical mitochondrial protein, and we did a radioactive assay for this, as well as to revisit the notions that synuclein is detectable at the level of mitochondrial preparations, and we did that through a range of cell and tissue. So first, what about the radioactive assay? This is the summary of our results. Simply to explain briefly what we found is that here we use as a positive control a canonical protein, NDH2, that stands for malate dehydrogenase 2. And as you can see, in normal situations where you have a normal membrane potential at the level of the mitochondria, the full-length protein or pre-protein, over time, it's processed or cleaved in these shorter fragments. If you eliminate the membrane potential, since this is a mechanism that is potential dependent, you see the full length, but you don't see anymore the process fragments. Now, if in these experiments we use also proteases like trypsin, what you see is that trypsin gain access to the full length, which remain outside the mitochondria, and digest it. But as you can see here, the process fragment, which is inside, is basically protected by the membrane of the mitochondria against these proteases, and you can detect it. What did we see for synuclein? A very bizarre pictures that it's not typical for a mitochondrial protein. The first thing we saw is that clearly we were able to detect radioactive synuclein at the level of the mitochondrial preparations, and these accumulations was clearly dependent of membrane potential. Yet we failed to detect a temporal increase of accumulations, and we did not see any evidence of processing. Now, if we put the trypsin experiment at play, we will see that in fact, yes, we have some digestions, but that was not a great digestions of the outside protein, suggesting that there are some elements that suggest that synuclein is indeed associated with the mitochondria, but on the other hand, some other features that made it absolutely atypical for a mitochondrial protein. About the Western blot, again, it was consistent with the association of synuclein in the mitochondria. So here we took cell line or human brain or mouse brain, and we prepared a protein extract that we basically split either in crude mitochondria or cytosolic fractions. And of course, we saw synuclein in cytosolic fractions everywhere, which we know that that's the normal distributions of synuclein. And confirming what many other studies have shown before, we also detected mitochondria in the crude mitochondrial preparations. Having said that, the crude mitochondrial preparations, it's a lot of fractions, particulate fractions of the cell. What it is, it's enriched in mitochondria, but it's not containing only mitochondria. And so among the different fractions, basically the inset here concentrate on only two, the MAM and the pure mito. And what it's striking here, as you can see, is that when we subfractionate these crude mitochondria fractions, everything is recovered in the MAM, and in fact, in the mitochondria per se, we don't see anything. What about comparing wild type to the pathogenic mutant of synuclein? Slightly different in the sense that, again, we found plenty in the MAM, but also the fact that the mutants accumulate in the pure mito, something that we didn't see in the wild type, suggesting that those point mutations may promote a redistribution between the MAM and the mito in this situation. We confirmed that by another technique, which is basically a sucrose gradient centrifugation that allow us to basically fractionate elements that it's enriched in what we call the detergent-resistant membrane, which correspond to the MAM. And then we can analyze what it's contained in these different fractions. And we use as a marker of the MAM, the flotilene, which is a typical marker of these DRMs. And what you can see is that very consistent with the Western blot data that I just showed you, there is plenty of wild type in the MAM, but much less of the two mutant. One of the questions we ask is whether the presence of synuclein wild type or mutant may affect the ability of the MAM to oppose to the mitochondria. So normally that's what we see, and we have a small distance that we can basically use as a proxy by colocalizations on confocal microscopy, as you can see here. And if you have a greater distance, for example, you see less of this. And that's exactly what we saw, in the sense that the wild type seems to have no effect on the oppositions of ER to the mitochondria, while the mutants, both of them, seems to reduce the ability of the ER or the MAM particularly to oppose to mitochondria. So structurally, it seems that the presence of the mutant, in fact, modified this element. As I told you before, the MAM has several functional elements. There is the synthesis of phospholipids that depend on it, regulations of calcium signaling homeostasis, and fusion, fission, and more specifically mitochondrial dynamics through DRP1 that all are localized and depend on the MAM. So let's see, therefore, what we can see in terms of impact on the functionality by the presence of wild type or mutant synuclein. And here, the first graph represents our results for the phospholipid synthesis. And what we can see is that indeed, in cells that overexpress the mutant synuclein, there is a reduction in the capacity of synthesizing phospholipid compared to the wild type, and very comparable data that I'm not showing, but that I'm citing for the calcium homeostasis. What about the mitochondrial dynamics? We assess that by the size of the mitochondrial dynamic, and what we saw is that, again, in those cells that overexpress either wild type or mutant synuclein, we had an increased frequency of fragmentations. In other words, smaller mitochondria, almost like DRP1, which is a MAM protein, would be hyperactive. So one of the ideas that we had was that because we have detected the fact that there is less mutant synuclein in the MAM and more in the mitochondria, perhaps the impact on the MAM integrity and functionality has to do to a loss of function. In other words, that those mutants, by quitting the MAM and going to the mitochondria, do not put enough synuclein in the MAM to assure proper functions. And so to test this idea, we basically overexpress, in addition to expressing the mutant, wild type to try to compensate, because if you remember the image with the flotilene, there's plenty of wild type in the MAM. And by doing so, as you can see here, we were unable to rescue the phenotype, suggesting that, yes, maybe there is an element of loss of function by this mutant to quitting the MAM, but that is not sufficient to explain the effect that we have detected. The next things we did was that to test the idea that perhaps the accumulations of synuclein mutant at the level of the MAM, it's stimulating DRP1, a mitochondrial dynamic protein. We use a specific inhibitor or nullifying the expressions. And in both cases, we saw no rescue of the fragmentation phenotype, partially, but not completely, suggesting that, yes, maybe the RP1 activities is dysregulated in the presence of mutant synuclein, but there seems to be other mechanisms that are driving this fragmentation phenotype. So in conclusion, what we have found so far it's interesting data that suggests a role for synuclein in the MAM function at different levels, but so far, those data were generated in very artificial system and that overexpressing a massive amount of synuclein. And so we thought that it would be interesting to try at least to confirm the salient point that I present to you by using human midbrain dopaminergic neurons and that express either no synuclein or endogenous level of wild type of mutant. And to do that, we basically turn our attention to iPS-derived dopaminergic neurons. And here is the results of our differentiations of the patient iPS. And in red, you have the dopaminergic neurons that we use to study further this question of synuclein and neurons. And so basically here it's simply to show you the different cell lines that we have generated. Here the mRNA, here the proteins. And as you can see from this Western blot, indeed, the knockout that we have generated by CRISPR-Cas had no detectable expression of synuclein. The control has nice level that were not significantly different from the A53 mutant. And indeed, the duplications had roughly 1.5 higher level. However, as you can detect, we had a problem with our A30P, and those are in the process of being repeated to see if those represent interesting data or rather artifact of our methodology here. This slide is simply to confirm that not only we saw synuclein in the MAM in many other cells, but in our iPS-derived dopaminergic neurons as well, as you can see here for the example from our control 18. And although there is still a lot of validations experiment and testing experiment that are ongoing, I can already report to you the fact that when we assess in those iPS-derived dopaminergic neurons that have or have not synuclein wild type or mutant, basically we see very similar alterations on the apposition frequency of ER to the mitochondria. And so what you see here compared to control is that the mutant is reduced, the multiplication is higher, that was consistent with the M17 data that I showed you before. But what is really interesting and striking and unexpected is the fact that when you have no synuclein, you also have higher apposition, very similar to duplications, suggesting that here you really regulate the MAM apposition, both when you have too much or too little of synuclein. So it's very important, therefore, I would suggest to have the right amount of synuclein at the level of the MAM to have a proper, at least structural organizations of these elements. So let me stop here and leave you with the following take-home messages. First of all, to stress the fact that wild-type synuclein, apart from being cytosolic, we have reported that it's also present in the MAM. The important aspect in relation to mutant is the fact that mutant synuclein seem to redistribute from the MAM to some extent to the mitochondria and perhaps to the cytosol as well. And we are in the process of determining if that's the case or not. Nonetheless, what we found is that the mutant, by altering some elements of the MAM, have functional impact on the MAM, which is the apposition frequency, the lipid metabolism and calcium transfer, but also on mitochondrial dynamic. On a biological point of view, we are excited about this data because, first of all, we think that if we correct, that may explain why this strange mitochondrial protein has been often associated with mitochondria through studies using crude mitochondrial preparations. And so basically, we believe that by fractioning further, it can be seen that it's not in the mitochondria per se, but rather in the MAM. And to some extent, I may even suggest a provocative ideas that the association or the loose associations of synuclein with at the nerve terminal, at the synaptic vesicle, may also reflect the proximity of MAM elements that exist in synaptic vesicle and at the nerve terminals, rather than really the synaptic vesicle per se. So with these studies, whether MAM and synuclein may have a role in pathogenesis, I would think so, but of course, many more work needs to be done. But certainly, I think that it opened a new way to see how synuclein and mitochondrial story may play into PD pathogenesis. Let me stop here, acknowledging the member of my lab, and particularly those who have contributed significantly on this work, Peter Barbuti, Cristina Guardia Lagarda, Estela Arias Gomez, and of course, our collaborator from the University of Luxembourg, Reiko Kruger, who is instrumental in all the different lines of IPS that we have used in this work. Thank you very much.